Hello students, welcome to the next lecture of antigen antibody reactions. In this lecture, we are going to start with enzyme linked immunosorbent assay, that is ELISA. It is one of the most popularly used antigen antibody reaction in diagnostic immunoassay. So let us look at the basics in the principle and the types of ELISA test. Since just to have a link with the previous lecture, we are learning various types of conventional and newer techniques of antigen and antibody reactions carried out in vitro for the qualitative and quantitative antigen and antibody uh, used in the diagnostic purpose in the serology. We have seen precipitation, agglutination, complement fixation reaction under conventional techniques. And in the last lecture, we have also seen radioimmunoassay. And in today's lecture, we are going to see enzyme-linked immunosorbent assay, that is ELISA. Now, basically, ELISA test is different than the radioimmunoassay in one of the major fact, that is it uses a reporter or a tag that in a way in a radioimmunoassay the tag used or a label used was a radioisotope, whereas in ELISA the reporter or a tag used is an enzyme. This is a basic difference in radioimmunoassay and ELISA. Apart from that, the radioimmunoassay can detect an antigen. It can detect and quantify an antigen, whereas ELISA can detect both antibodies and antigen in the blood. Both your radioimmunoassay and ELISA are highly sensitive. So even ELISA is very sensitive, and basically it depends on monoclonal antibodies. I'm sure you have gone through the video of primary and secondary antibody, which I have passed on to you and given you as a prerequisite for watching before listening to this lecture. So if not, I kindly request you to stop this lecture here itself. Go to the prerequisite video of this lecture, that is primary and secondary antibodies. Complete that assignment and then come back to this lecture. Because without that prerequisite, you won't be able to understand this lecture. So to continue, there are three types of ELISA test. Direct ELISA, which is basically used for detection of antigen. Indirect ELISA, one of the most popular for detection of antibody. And sandwich ELISA, which is for the detection of again antigen. We are going to learn all the three types of ELISA test in today's lecture. Sample specimen used in ELISA test can be serum, it can be CSF, it can be sputum, it can be urine, it can be stool. Depending upon the sample serum, you can see depending upon what is the purpose of diagnosis, you use a different kind of sample. For example, for HIV, you use a serum. For neurocysticercosis, you use CSF. For brucellosis, you use sputum, and so on. So depending upon what is the purpose, what exactly you want to find out, depending upon that, your sample changes. Then let us look at the basic mechanism of ELISA. Whatever is the type, direct, indirect, sandwich ELISA, basic mechanism which we will try and correlate, since we have learned in a last lecture radio immunoassay. Let us try and correlate the basic mechanism of ELISA and learn what exactly the ELISA test is all about. So as I mentioned, ELISA or radio immunoassay is again based on an antigen and antibody reaction. So obviously there is one antigen and there is an antibody. And antigen and antibody, they are going to interact and give you a reaction. We have seen in the last lecture, these all immunoassay are used when antigen and antibody are reacting, but they are reacting in such a low concentration that we are not able to detect them. And for detecting such a low concentration, which is not visible, not giving precipitation and agglutination, this reaction is detected by adding it. In case of ELISA, this label is enzyme. So when antigen is reacting with the antibody, this reaction in ELISA is detected by enzyme. By adding an enzyme, obviously, this enzyme needs to be reacted with certain substrate. And when this enzyme reacts with the substrate, this enzyme substrate reaction is going to generate a product, and this product is colorful. And because of this colorful product, we get an indication that antigen and antibody has reacted and the test is positive. This is a simple mechanism. So let us begin to understand the mechanism once again. So antigen reacts with the antibody, which is enzyme labeled. Enzyme used over here are largely of two types: horseradish peroxidase and alkaline phosphatase. So I repeat, the enzyme which are attached to the antibody, which are labeled, are horseradish peroxidase and alkaline phosphatase. Unlike radioimmunoassay, the labeling is done to the antibodies in the ELISA. In radioimmunoassay, the antigen were labeled. In ELISA, the antibodies are labeled. So the labeling is done with the enzyme either horseradish peroxidase or alkaline phosphatase. And these enzymes are going to react with certain substrate. So these substrates are, if the enzyme uses horseradish peroxidase, then the substrate can be O-phenylene diamine dihydrochloride. If enzyme uses alkaline phosphatase, then substrate is p-nitrophenyl phosphate. I hope I'm clear. Then let us understand the mechanism. When antibody, which is labeled with the enzyme, attaches to the antigen, then this antigen antibody reaction has happened, can be detected by this enzyme. The moment this reaction happens, this you can add the substrate to detect the reaction. The substrate reacts with the enzyme and converts into product. And this product develops a color, and this colorful reaction, which is also called as chromogene, and this colorful reaction indicates the test is positive. This is a basic mechanism of ELISA. So let us see now the direct ELISA. The first type of ELISA is direct ELISA. We have three types: direct, indirect, sandwich. We are going to look at one by one each. So direct ELISA, the purpose of direct ELISA is to detect the presence of antigen in a sample. Now, microtiter well is coated with the antigen. I have just shown here the 96 well plate. When we always say in the lecture microtiter well, then you must understand what we are talking about. This is a 96 well plate. 96 well plate means this is a plate in which there are 96 well. So when I am saying microtiter well, then I am representing one of the well into it. So this line indicates one of the well into it. Now, so microtiter well is coated with the antigen. Why? Because the direct ELISA detects the presence of antigen. तो जो भी एंटीजन डिटेक्ट करना है that is coated into the well. Obviously, if antigen is coated, then the specific to that antigen, the antibody is added. And this antibody is enzyme linked. This antibody is enzyme linked. This antibody is specific to the antigen, so it will go and bind to the antigen. And since this is enzyme linked, after this step, you will add a substrate to this, and this enzyme will convert the substrate to the color product, and you will get the color, and you will understand the reaction. The color will indicate there is a presence of antigen.